Hi there and welcome to PhD at Living. Today we're going to continue our discussion about how humans see color. From our last video, we know there are three types of cone cells in the human eye, and each type is responsible for a certain piece of that 400 to 700 nanometer range of the electromagnetic spectrum that humans can see. Now we're going to see, get it? What happens when something goes slightly awry. There are two things that can happen to a given cone cell. It can either malfunction in some way or not function entirely. This leads to a variety of types of color blindness. Let's take the really drastic scenario first. If all three cone cells are completely non-functional, the only way a human can convert light into vision is with their rod cells. However, we know the rod cells don't see color, so a person won't have any color vision or be able to distinguish any hues, and can really only tell the difference between intensity of light. This is called rod monochromacy and is extremely rare. In addition, rod monochromacy comes with really poor vision, talking like 20/200. Moving up a level, we get to cone monochromacy. Here, one cone is functioning and the other two are dead. Remember that the brain uses an opposite process of comparing the cone's signals to one another in order to determine what color is being seen. With cone monochromacy, because only one cone is functioning, the brain doesn't have anything to compare it to. Hence, the person's colorblind. The consolation prize with cone monochromacy is the person will usually have normal vision and can distinguish some hues from one another. Next, we have three types of dichroism. That is, two cones are functioning and only one is dead. This is what we usually think of when we talk about classical color blindness. Our first two dichroisms are protanopia, or red color blindness, where the L cone is missing, and deuteranopia, or green color blindness, where the M cone is missing. They're pretty similar, and that's why we're going to talk about them together, and they both live on the X chromosome, meaning males are more likely to have these conditions. Hot little biology intersection here. A person usually gets a single unit of each chromosome from each of their parents. For a simple dominant recessive gene in a given chromosome, that expresses one of four ways. Big D, big D, big D, little d, big D, or little d, little d. For the little d recessive trait to be expressed, that child needs the recessive trait from both of their parents. If either one of the parents gives that child the dominant big D trait, whether through big D, little d, or little d, big D, the child will not express that recessive trait. As it turns out, the red and green color blindness genes are recessive, and they live on the X sex chromosome. Because males get a Y chromosome from their fathers, they only have one chance of getting an X chromosome that has that dominant color blindness gene. If they get the recessive gene, they'll be colorblind. Females get two chances because they have two X chromosomes, which is why males are statistically much more likely to be colorblind than females are. A female who has normal vision herself but gives color blindness to her son is called a carrier, being that she carries the gene but it isn't expressed. Because the M cone and L cone overlap so much in normal vision, and because the brain uses the opposite process to figure out color, protans and deutans, the name for people who have protanopia and deuteranopia, tend to see the same degrees of colors. While protanopia and deuteranopia are usually called red-green color blindness as a blanket term, protans and deutans have troubles with reds, greens, and oranges, yellows, and browns. Deutans, who have a deficiency in the M cone, are by far the most common type of colorblind people. Our last type of dichroism is tritanopia. It's extremely rare and where the S cone is missing. This blue color blindness causes greens to be seen as a shade of blue and yellows to be seen as almost a shade of violet. The tritanopia defect lives on a gene on chromosome 7, so it's equally likely to hit males and females, although again, it's quite rare. There are also minor levels of color blindness where a cone is just malfunctioning and not completely missing. You can think of this in a couple different ways. For instance, let's look at this green M cone here where the intensity is dropped because the cone just isn't as sensitive to those wavelengths as it should be. Or we can look at something like this where the green cone spectral range has shifted and is now overlapping the red L cone much more than it should be. Either one of these or a combination or a different one of minor color blindness is significantly more common than a complete cone missing. The vision of people here is going to be somewhere between those of the truly colorblind and regular visioned folk. Let's go back to that spectral shift thing. You ever see those crazy glasses that make colorblind people see real colors for the first time? Oh, it's so beautiful, Baxter! Okay then. Well, it turns out they filter out a number of wavelengths. If we compare this normal spectral range here to this deuteranomalous one here, deuteranomaly meaning there's a problem with the green M cone, but it's still here, we can see the green cone is shifted over, almost completely overlapping that red L cone. Almost certainly this person's going to have a decent amount of green color blindness. Well, what the glasses do is they come in and they filter out a bunch of these wavelengths where that overlap between the green M cone and the red L cone happen. Therefore, the person never even sees these wavelengths, which, by the opposite process we know the brain does, allows the brain to see the other spectral ranges, other wavelengths that don't overlap, and be able to make a better judgment and give that person a more accurate, more realistic, better color. That being said, I think the glasses are only good for those types of anomalies, either a red protoanomaly or a green deuteranomaly. If the cone is completely missing, I don't think the glasses are going to help. In addition, with that S blue cone, if there's something going on with that bad boy, because its spectral range is so far from the green M cone and the red L cone, I don't think the glasses are going to help. Could be wrong though. Finally, and because I can't ever let anything go after I see it, we're going to talk about rods and night vision. I've put up here, again, the figure of intensity versus wavelength for each of the 3 cone cells, but I've also put the spectral range for the rods. The rods I've drawn in black because, again, rods don't see color, but they do get light and contribute to vision. What we're talking about here is as darkness approaches, the rods, being more sensitive than the cones, start to dominate our vision. In addition, because the rod's intensity is highest at about a wavelength of 500 nanometers or so, the colors around that wavelength start to get accentuated. On the flip side of that, you can see the rods don't have much intensity at all outside of 600 nanometers or so the reds start to fall away. As it's very dark, approaching almost complete darkness, and color goes away entirely, reds start to look much darker, maybe even black, and blues really start to pop. This is called, depending who you ask, the Purkinje or Purkinje Effect, and it's named after Czech physiologist Jan Purkinje. Pretty cool, no? And that's the chemistry of color blindness. Next time we're going to talk about the vision of some game animals and man's best friend. But that's for then, and this is for now. See you next time. No way. No way. Tried it once, it doesn't work. You get 4 guys all fighting over who's going to be Mr. Black, but they don't know each other, so nobody wants to back down. No way. I pick, you're Mr. Pink.